This conference will now be recorded.
Hello students. So for NEET 2024 we are preparing now, and here we are in the 13th chapter, which is coming under botany. So we are learning about photosynthesis in higher plants, and here you have to learn uh, about almost all the processes which are involved in the photosynthesis of higher plants. See, that means photosynthesis in the aspect of autotrophic nutrition you need to learn, and the site of photosynthesis. Then next, the pigments which are involved in photosynthesis. Like, you are getting almost all the details of the process of photosynthesis. Then biochemical and photosynthetic phases of photosynthesis, what is cyclic and non-cyclic photophosphorylation, chemiosmotic hypothesis, photorespiration, then C3 and C4 pathways, then which are all the factors which is affecting photosynthesis. About all these concepts we are learning here. And here in the previous part we had learned about the site of photosynthesis, then the pigments which are involved in photosynthesis, and the mechanism of photosynthesis. Here we need to start with the mechanism of photosynthesis and the process of photosynthesis, that is divided into two phases. So process of photosynthesis, here two phases we can see. Which are all the two phases in photosynthesis? That is divided into photochemical phase and biosynthetic phase. Photochemical phase, and the second one is biosynthetic phase. Here in the photochemical phase, or this is the light reaction, we said light reaction, or we can say Hill reaction. Okay, so the see here in this chapter, the photosynthesis, in this chapter, here I am marking the main points which are the repeated questions. Okay, so photochemical phase, that is otherwise called as light reaction or Hill reaction. So Hill reaction, this is one repeated question of previous year questions. In previous year questions you can see this question, Hill reaction, that is the photochemical phase, and that is the light reaction. So this is taking place inside thylakoids. So let's see about this photochemical reaction. So this is inside the thylakoids, or we can say the grana of chloroplast. Grana of chloroplast. Okay, and here the function of this phase, that is, what is the function, main role of this phase, that is producing assimilatory powers, that means production of ATP, NADPH, production of ATP, NADPH. So production of all these assimilatory powers, that is the main function of this phase, photochemical phase. And see, during this production, what and all processes are involved in this? Here are the events, which is like light absorption is taking place. Light absorption is there. Then water splitting, splitting of water. Then release of oxygen. Then here high energy chemical intermediates are forming. So all these things are taking place during the light reaction or Hill reaction or the photochemical phase. Light is getting absorbed, water is getting splitted, oxygen is getting released, then chemical intermediates are forming in this phase. So all these are taking place, and several protein complexes and so many pigments are involved in light reaction or photochemical phase. So here pigments are involved, light reaction in this, pigments are involved, several complexes of protein and the pigments, those are involved in this. Which one? Light reaction. Okay, so here we said about the light absorption, right? So light absorption, so the molecule which is responsible for the absorption of light absorbing molecule, that are protein based complexes. Okay, so that's a protein based complex which is called as light harvesting complex, or LHC. In short form we can write as LHC. So these light harvesting complex, they are organized into photosystem 1 and photosystem 2, PS1 and PS2. Okay, so photosystem 1 or pigment system 1, that also we can call photosystem 1 and photosystem 2. In this, these light harvesting complexes are organized. So photosystem 1 or pigment system 1, here the reaction center in this pigment system is P700. Okay, so here that absorbs the light of wavelength of 700 nanometer. That's what by 700 we are mentioning here in photosystem 1 or the pigment system 1. The reaction center in this pigment system is P700, which absorbs the light of wavelength 700 nanometer, and PS1 or photosystem 1 can carry on the cyclic photophosphorylation independently. Here independently, that can carry the cyclic photophosphorylation. Okay, which one? Photosystem 1 can carry the cyclic photophosphorylation independently. The photosystem 1 with electron carriers, that is located on both the non-appressed parts of granathylakoid and stromathylakoid. Clear? So these are the points which we need to mention. These all the points which I said now. Here, light absorbing molecule, light harvesting complex, that is photosystem 1 and photosystem 2, that is organized into photosystem 1 and 2. So you should know LHC, PS1 and PS2. And here in PS1, pigment system 1, reaction center is, that is P700. That means the wavelength of the light, that will be 700 nanometer. And in photosystem 1, that can carry on the cyclic photophosphorylation independently. That is another important repeated question. Okay, so PS1, photosystem 1 or pigment system 1 with electron carriers, that is located on both the known abreast parts of stromathylakoid and granathylakoid. So these are the main points, and from here only you will get question in the photosynthesis. Okay, or the about PS1. Now you have got all the points. Next we are moving to PS2 or photosystem 2 or the pigment system 2. Here in the pigment system 2, P680, that functions as the reaction center. This is the reaction center. That means the photons of lower wavelength, that is 700, PS1 is 700, photosystem 2 is P680, or the reaction center is P680 is functioning as the reaction center in pigment system 2. So the photon of lower wavelength, here lower wavelength, it is coming to 680, right? So it's of lower wavelength and that is absorbed by this photosystem 2. So this is located in the appressed part of thylakoid, that is non-appressed part in PS1. Here in the appressed part of thylakoid. And here you need to mention a very important point, that here this can carry out non-cyclic photophosphorylation with PS1. Non-cyclic photophosphorylation. Photophosphorylation with PS1. Okay, that one, PS1 is independently, it can carry out the cyclic photophosphorylation, right? But here in PS2, with the help of PS1, this can carry out non-cyclic photophosphorylation. Okay, so that's about the light absorption. Next, we said about splitting of water, right? This processes are the events in photochemical phase, that is light absorption. Light absorption, PS1 and PS2 now we have learned. Next is splitting of water or photolysis of water. Splitting of water, or we can say photolysis of water and release of oxygen. And release of oxygen. These are another two events which are happening in the photochemical phase or light reaction. Okay, so in photosynthesis, the water is used as a source of hydrogen which is required for the reduction of carbon dioxide. Okay, this is one repeated question. Role of water. So in photosynthesis, water, H2O, this is used as a source of hydrogen. Source of hydrogen which is required for the, and this hydrogen, what is the role of this hydrogen? This is required for the reduction of carbon dioxide. Reduction of carbon dioxide from carbohydrate. Carbohydrate. Okay, so here you are getting 3 points. Hydrogen is the source of, sorry, water is the source of hydrogen, and why it is required, this hydrogen is required for the reduction of carbon dioxide, and that forms the carbohydrate. So this breakage of water, this photolysis or the splitting up of water, that is catalyzed by manganese. Okay, so manganese, there is a role of for this manganese in this reaction. So breakage of water, breakage of water, that is catalyzed by manganese. Okay, and this is in the form of manganese Schiff base. Manganese Schiff base. Okay, this is very, very, very important, and this is a repeated question. Here I'll write the reactions: carbon dioxide plus 2 water molecules, 2 H2O in the presence of light. This is a light reaction, right? And here pigment is also there, chlorophyll. So what you will get, CH2O plus H2O plus O2. Here oxygen is getting released, right? And 4H2O in the presence of light and chlorophyll. Here 2H2O plus 4H plus. Here oxygen, this hydrogen, source of hydrogen, this hydrogen is required, right? And 4 electrons plus oxygen. So this is the reaction. Here the splitting of water is taking place in this way. So here you are getting several questions. Water is the source of hydrogen, and what is the role of hydrogen for the reduction of carbon dioxide, and that forms the carbohydrate. So the breakage of water, that is catalyzed by which ion? Manganese, and that is in the form of manganese shift base. Okay, and these two reactions you need to keep in mind. Okay, next, formation of higher energy chemical intermediate we said, right? First one we said light absorption, then splitting of water, release of oxygen. Now splitting of water and release of oxygen, both we have seen. Next is the formation of high energy chemical intermediates. Formation of high energy chemical intermediates. So here, the formation of high energy chemical intermediates, they are reduced molecules which provide energy during biosynthetic phases. This high energy chemical intermediates provide energy. That's why we are saying high energy chemical intermediates. They are providing energy during the biosynthetic phase. During biosynthetic phase. See, photochemical phase is there and biosynthetic phase is there. But here the formation of high energy intermediates, here it is forming. This high energy chemical intermediates are forming, and this is providing energy during the next phase, the biosynthetic phase. Okay. So these intermediates are NADPH2. Which are the intermediates? NADPH2. This is high energy chemical intermediate, and NADPH. NADPH. These are the chemical intermediates. Then we can say ATP. All these are the high energy chemical intermediates. Okay, and all these are produced by two types of reaction. One is photophosphorylation. These are produced by two types of reactions: photophosphorylation and chemiosmotic hypothesis. First one is photophosphorylation, and second one is chemiosmotic hypothesis. Chemiosmotic hypothesis. Okay, so let's see first the photophosphorylation. So all these points you need to note down. This question, this high energy biosynthetic phases, which are all the high energy chemical intermediates which are helpful in the biosynthetic phase. So these is important repeated question, and two types of reactions are taking place for the formation of these. So which are the reactions? Photophosphorylation and chemiosmotic hypothesis. These are repeated questions. So let's see one by one. Photophosphorylation. Photophosphorylation. This photophosphorylation, that's the formation of ATP molecule. Formation of ATP molecule from ADP and H3PO4. Okay, so this is taking place. All these are coming in the light reaction, right? So this is in the presence of light and chlorophyll A. Presence of light and chlorophyll A. So this is repeated question, and this is also a previous year question. And this process, formation of ATP from ADP and H3PO4 in the presence of light and chlorophyll A, that is called as photophosphorylation. So we'll write as ADP plus H3PO4 in the presence of light and chlorophyll A, that forms ATP. Okay, so this reaction, this also you should remember, because they'll give a reaction and this indicates which step of this photosynthesis. Then they'll give the options. So like that also you can expect question. One time it came, this question came. So ATP is getting formed like this. Okay, so ATP formation, that takes place through two types of phosphorylation reactions. Okay, ATP formation, that is taking place through two types of phosphorylation reaction. First one is non-cyclic photophosphorylation. First type is non-cyclic photophosphorylation. Non-cyclic photophosphorylation. And second one is cyclic photophosphorylation. Cyclic photophosphorylation. So ATP is getting formed in these two ways, cyclic photophosphorylation and non-cyclic photophosphorylation. Okay, so in the non-cyclic photophosphorylation, both ATP and NADPH2, ATP and NADPH2, both ATP and NADPH2 are produced in non-cyclic photophosphorylation. Okay, during non-cyclic photophosphorylation, initial donor and final acceptor of electron, initial donor of electron and final acceptor of electron, both are different. In which one? In non-cyclic photophosphorylation, you can say that ATP and NADPH2 are getting produced. So the initial donor of electron and the final acceptor of electron, both are different. Okay, and after the illumination of PS2 or photosystem 2, after the illumination of PS2 or photosystem 2, the released electron passed to PS1. Released electron passed to PS1 through different electron carriers. Okay, and from PS1, So from PS2, that electron which is getting released, that is passing to PS1 via various electron carriers. And from PS1, electron is finally provided to ferredoxin. From PS1, finally that electron is provided to ferredoxin, or we can write FD, ferredoxin, and that help in the synthesis of NADPH2 from NADP. This ferredoxin helps from photosystem 1 or the pigment system 1, electron is finally given to ferredoxin, and this is helping for the synthesis of NADPH2 from NADP. This is called as Z scheme. This is called as Z scheme. So this, why we are calling a Z scheme? Due to the characteristic shape. Okay, the process, due to that characteristic shape, it is called as Z scheme. Okay, so that's the main points about the non-cyclic photophosphorylation. Okay, so here the final donor, final acceptor and the initial donor of electron, both are different. So after illumination of PS2, when electron is getting released, that will pass to PS1 through various electron carriers. And then from PS1 or the photosystem 1, electron is finally provided or given to ferredoxin, and that helps in the synthesis of NADPH2 from NADP. So this, due to the characteristic shape, it is called as Z scheme. Okay, all these are previous year questions. Okay, from previous year questions, I'm selecting and giving the points. Okay, next about the cyclic photophosphorylation. So in cyclic photophosphorylation, the process, in that process, initial donor and the final acceptor of electron is the same. Initial donor and final acceptor of electron, both are same. Okay, in the cyclic photophosphorylation, don't get confused. In non-cyclic, both are different. In cyclic, initial donor and final acceptor of the electron, that is the same chlorophyll A. Which one is that? Chlorophyll A of photosystem 1, PS1. Okay, so here both are same. Then what is that? Which one is accepting that electron? Chlorophyll A of photosystem 1. So only photosystem 1 is involved in this phosphorylation. Okay, only PS1 photosystem 1 is involved in cyclic photophosphorylation. We said a photosystem 1, the cyclic photophosphorylation, that is independently carried out by which one? Photosystem 1 or the pigment system 1, and there that absorbs the light wavelength of 700 nanometer. And we said PS1 can carry on the cyclic photophosphorylation independently. So this is the reason here. Initial donor and the final acceptor of electron both are same. There is no involvement of PS2. And which is that final acceptor and initial donor? Chlorophyll A of photosystem 1. So there is no role for the photosystem 2 in the cyclic photophosphorylation. Clear? And it occurs in stromal lamellae. Stromal lamellae. There it is taking place, and the process of cyclic photophosphorylation, we know that occurs when activity of PS2 is ceased. Okay, when the cyclic photophosphorylation is taking place, some important points: when the activity of photosystem 2 is ceased, activity of PS2 is ceased or stopped. non-cyclic photophosphorylation stops due to some reasons. Okay, due to some reasons, the non-cyclic photophosphorylation is getting stopped. So that occurs when that time only the cyclic photophosphorylation is taking place. Okay, so the electron emitted after illumination of PS1 that returns back to the original place through several electron carriers, and which ultimately lead to the synthesis of NADPH. Okay, so this is the cyclic photophosphorylation and non-cyclic photophosphorylation, which is coming under photophosphorylations. Okay, one is cyclic and non-cyclic. Next, we need to say about the chemiosmotic hypothesis. So the formation of high energy chemical intermediates, we said these intermediates like NADPH2, NADP, ATP, all these are formed by different types of reaction. One is photophosphorylation and another one is chemiosmotic hypothesis. We have already learned about photophosphorylation. In that, non-cyclic photophosphorylation is there, cyclic photophosphorylation is there, right? Next is about the chemiosmotic hypothesis. Chemiosmotic hypothesis. So through this process also, you can see there the formation of high energy chemical intermediate is taking place. Okay, so chemiosmosis, we are saying about the chemiosmosis in chloroplast. Okay, so this explains the mechanism of how actually ATP molecule is synthesized in the chloroplast. ATP molecule synthesis in chloroplast, that we can understand from the chemiosmotic hypothesis. Okay, so how ATP molecule is synthesized in the chloroplast, that's what we are learning in chemiosmotic hypothesis. So in brief, we can say chemiosmosis requires a membrane, a proton pump, proton gradient, and ATPase, the enzyme. Okay, so chemiosmosis, what are all the required condition for this chemiosmotic hypothesis? That is a membrane, then a proton pump, proton gradient, and ATPase enzyme. Okay, so this ATPase that has a channel which allows the diffusion of protons back across the membrane. Which one? ATPase is having a channel. This channel allows the diffusion of protons back to the membrane, which is leading to the breakdown of gradient. Okay, and this releases energy, or enough amount of energy is getting released to activate the ATPase enzyme, and that catalyzes the formation of ATP. So this proton gradient is getting broken down. So here the energy is getting released, and this energy is more than enough to activate which one? ATPase enzyme. ATPase enzyme. There an activation energy is required to make it activate, right? So if the energy, that released energy is getting utilized to activate ATPase enzyme, what is happening there? That catalyzes the formation of ATP. Formation of ATP. This catalyzes the formation of ATP, and this ATP, so finally after catalyzing that reaction, ATP is getting formed. So that ATP is used immediately for the biosynthetic reaction. ATP is used for biosynthetic reaction, which is the next step, right? First is the photochemical phase, and next that ATP which is formed, that is used for the biosynthetic reaction which is taking place in the, where this biosynthetic reaction is taking place, that is in the stroma, which is responsible for fixing carbon dioxide and synthesis of sugar. Okay, so the products of light reaction, what are all the products of light reaction? See, this is very important point. Products of light reaction, and this is most repeated question. The products of light reaction, that is ATP, NADPH, and oxygen. And here we said oxygen. This oxygen, that is diffuse, oxygen diffuses out of the chloroplast. Oxygen diffuses out of the chloroplast, but ATP and NADPH, they are used to derive the process which is leading to the synthesis of food or sugar. Here ATP and NADPH. Second point, ATP and NADPH. The products are getting formed. So we should know what is happening to these products. Oxygen. First we are saying about oxygen that diffuses out of the chloroplast. And what is happening to ATP and NADPH? This ATP and NADPH, they are used to derive the processes which is leading to the synthesis of food. Synthesis of food, or we can say for the synthesis of sugar. Clear? So this is the photochemical phase or light reaction or Hill reaction. Okay, so under photosynthetic phase we are learning about light absorption, photolysis of water, then formation of high energy chemical intermediates. So in the high energy chemical intermediate formation, we are learning about photophosphorylation and chemiosmotic hypothesis. In photophosphorylation, cyclic and non-cyclic photophosphorylation, we are learning. Then chemiosmotic hypothesis means there the mechanism of ATP molecule synthesis in the chloroplast. That's what we are learning. Okay, so that's all about the light reaction or Hill reaction or photochemical reaction. Next, we need to learn about biosynthetic phase. So light reaction over. Next is dark reaction of photosynthesis. Biosynthetic phase. Biosynthetic phase, or we can say dark reaction or black man's reaction. This is very important. Black man's reaction, that is a repeated question. Dark reaction or biosynthetic phase or black man's reaction. All the three names you should remember. So this biosynthetic phase or black man's reaction or dark phase, dark reaction, this occurs in the stroma of chloroplast. Occurs in the stroma of chloroplast. Okay, it occurs in the stroma of chloroplast, and this process does not directly depend on the presence of light. There is no direct light dependent process in dark reaction. That's why from the name itself you can make it out. So in that way you should remember dark reaction means there is no directly dependent light events. So light is not directly involved. There is no reaction in this which is directly depend on the presence of light, but it is dependent on the products of light reaction. Okay, no events are light dependent, but But here, all the light reaction products, it is exactly, or it is directly depending upon the light reaction products. Depends upon products of light reaction. Right, that high energy intermediate chemical intermediates, all these are getting utilized here in the biosynthetic pathway, right? So here, these products of light reactions are completely utilized. That means ATP, NADPH, then carbon dioxide, water, all these things are getting utilized in dark reactions. So there are three different pathways for carbon dioxide fixation in plants. Okay, three different pathways for carbon dioxide fixation, CO2 fixation in plants. First one is C3 pathway. C3 pathway or Calvin cycle. C4 pathway. Second one is C4 pathway or Hatch-Slack cycle. Third one is CAM, crassylation acid metabolism. CAM pathway, crassylation acid metabolism or CAM pathway. Okay, these are the three different pathways for carbon dioxide fixation in plants. Okay, so let's see one by one. Now, C3 pathway or Calvin cycle. C3 pathway or Calvin cycle. Let's see the main points. See, these points are mainly, all the points are from previous year questions. So after learning this, you can answer all the previous year questions. So here the C3 pathway or Calvin cycle, this was discovered by Melvin Calvin. Melvin Calvin. All the points are prepared from the previous year questions. And this explains the process of carbon dioxide fixation in green plants. Okay, C3 pathway that explains the carbon dioxide fixation in green plants, and in this pathway the assimilatory power or the NADPH or NADP and ATP, all these produced in light phase, all these are getting used to reduce carbon dioxide. These are getting produced during light reaction. So all these are getting utilized to reduce carbon dioxide to carbohydrate. Okay, so C3 pathway that involves three major steps. C3 pathway steps that involves three main steps. Which are they? First one is carboxylation. Carboxylation. That means ribulose 1,5 biphosphate. Ribulose 1,5 biphosphate phosphoglyceric acid. Phosphoglyceric acid. Second one is reduction. That means now phosphoglyceric acid is formed. This phosphoglyceric acid to phosphoglyceraldehyde. Phosphoglyceric acid to glyceraldehyde. This is the second step. And in the third step, synthesis of sugar. Synthesis of sugar. In this, hexose sugar that is getting synthesized, or regeneration of RUBP. Regeneration of RUBP. That is the third step. So all these are the steps. C3 pathway involves three major steps. First one is carboxylation, then reduction, then the synthesis of sugar. Carboxylation means ribulose 1,5-biphosphate is, from that it is forming phosphoglyceric acid, and next is reduction. Phosphoglyceric acid is getting reduced to phosphoglyceraldehyde, then the synthesis of sugar, hexose sugar. Okay, or we can say the regeneration of RUBP from PGAL to N cycle. Okay, so in this cycle, 6 molecules of carbon dioxide, 6 molecules of carbon dioxide that is used, and 1 molecule of fructose 6-phosphate is produced. 1 molecule of fructose 6-phosphate is produced. Fructose 6-phosphate. That is produced as a byproduct at the, this is the byproduct. Okay, which is the byproduct, that here how many carbon dioxide molecules are getting used? 6 molecules of carbon dioxide is used, and 1 molecule of fructose 6-phosphate, that is the byproduct. And how many molecules of NADPH? Here 12 molecules of NADPH and 18 molecules of ATP. It is getting utilized. 18 molecules of ATP getting used. So the overall reaction, we can see that, so here this is the total expense of energy, 12 molecules of NADPH and 18 molecules of ATP. So the total reaction, how we will write the total reaction? 6 RUBP plus 6 carbon dioxide. Here we are writing, right? And 12 NADPH plus 12H plus again 18 ATP plus 6 H2O, that gives 6 RUBP. Here regeneration of RUBP we are saying, no, the third step, regeneration of RUBP. So after the reaction, 6 RUBP plus glucose, hexose sugar that is forming, plus 12 NADP plus 18 ADP plus 18 PR. Okay, and it's better you make a diagrammatic representation of the C3 pathway, and there you can write from which product next, which is the next step, carboxylation, reduction, and how many ATPs, and what is the energy consumption in this step. Like that you can easily mark. Okay, otherwise this point wise, this is the previous year questions. Okay, all the points I made from previous year question papers. So it will be easy for you. If this is easy, you can make like that. Otherwise, the schematic pathway you can make and mark in each step what is the changes happening in each step. Okay, so the diagrammatic representation of Calvin cycle you can make, and the regeneration of RUBP you can mark there, and the total expense of energy you can mark there. Clear? So in whichever way is easy for you, you can do in that way. Second pathway for carbon dioxide fixation in plants, that we can say the C4 pathway. C4 pathway. C3 pathway we said that is Melvin Calvin. Calvin cycle we can say that is discovered by Melvin Calvin. And here the C4 pathway or Hatch and Slack cycle. Okay, this second name also you should. C3 is Calvin cycle, C4 Hatch Slack cycle. So this was worked out by Hatch and Slack. Worked out by Hatch and Slack. Okay, so that's why it is also called as Hatch and Slack pathway. And the first product of carbon dioxide fixation in C4 pathway is a C4 acid, oxaloacetic acid. First product of CO2 fixation, carbon dioxide fixation in C4 pathway, that is oxaloacetic acid. So this is a previous year question. Oxaloacetic acid, OAA, that is the first product of carbon dioxide fixation in C4 pathway. And the C4 pathway occurs in plants like maize, sugarcane, sun plant. In all these type of plants, it occurs, C4 pathway. And in these plants, there is a special Kranz anatomy. Kranz anatomy. That means in this mesophyll cells are adjacent to the bundle sheath cell and which is containing large chloroplast. Okay, so here C4 pathway is taking place in maize, sugarcane, sun plants, etc., which is having Kranz anatomy. And Kranz anatomy means in that mesophyll cells which are adjacent to the bundle sheath. To the bundle sheath, near to the bundle sheath, mesophyll cells which are present, that is having or that is containing large chloroplast. Okay, mesophyll cells with large chloroplast. Clear? And all the known C4 plants, they are angiosperms. Clear? Here, the CO2 combines with 3-carbon phosphoenol pyruvate. Phosphoenol, 3-carbon phosphoenol pyruvate, PEP, in the mesophyll cells, and that forms oxaloacetic acid. That forms oxaloacetic acid, which is a 4-carbon compound, and malic acid. Okay, and this is then transported to the bundle sheath cell where the carbon dioxide is released to go into Calvin cycle. This will go to bundle sheath, and there the carbon dioxide is getting released to go to the Calvin cycle. Carbon dioxide will go to the Calvin cycle or the C3 pathway. Okay, here again, here also 30 ATP, 30 ATP and 12 NADPH2. 12 NADPH2. These are required for the formation of one molecule of hexose sugar or glucose. Okay, for the formation of 1 molecule of hexose sugar, here the energy they need to spend, that is 30 ATP plus 12 NADPH2 for 1 molecule of hexose sugar. So this is repeated several times. So in C4 pathway, what is the energy expense? And in C3 pathway, what is the expense? Everything you need to learn. And next is about CAM, another pathway which we said crassylation acid metabolism, CAM pathway. Third type is CAM pathway, which is the crassylation acid metabolism. So this pathway is mostly present in the succulent xerophytes. Present in succulent xerophytes. Xerophytes, like we can say that crassulaceae, euphorbiaceae and all. The members of, this is also CAM pathway where and all you can see members of euphorbiaceae and crassulaceae. Right. So in this process, during night time, stomata remains open. In CAM pathway, during night time, stomata will be open and carbon dioxide enters through that. Then through this open stomata, carbon dioxide enters. Okay. So this is accepted by, this carbon dioxide is accepted by oxaloacetic acid, and then it is getting converted into malic acid. Okay, during night time stomata will be open, through that carbon dioxide enters, and that is getting, or that is accepted, that carbon dioxide is getting accepted by oxaloacetic acid, and it is getting converted into malic acid. Okay, so at night stomata are open, our carbon dioxide is fixed by the action of, here through open stomata carbon dioxide enters. So this carbon dioxide is getting fixed by the action of phosphoenol pyruvic acid, is fixed by phosphoenol pyruvic acid carboxylase. Phosphoenol pyruvic acid carboxylase, PEP carboxylase, and malic acid is stored in the cell vacuole, and malic acid is stored in the cell vacuole. So during daytime, the stomata will become closed. So carbon dioxide is released from the malic acid, and that is fixed by Calvin cycle. So nighttime carbon dioxide enters. During daytime, stomatas are closed. So carbon dioxide that is released from the malic acid, and that is fixed by Calvin cycle. Okay, the C3 cycle or the Calvin cycle. Here during daytime, that malic acid we said that produced at night. Malic acid is getting produced at night time, right? So during daytime, the malic acid which is produced at night, that results in the production of both pyruvic acid and carbon dioxide on decarboxylation, right? So pyruvic acid and carbon dioxide on decarboxylation. So the pyruvic acid, that's how pyruvic acid is getting produced, and so produced pyruvic acid is recycled back to phosphoenol PEP. Okay, phosphoenol pyruvic acid. When that carbon dioxide enters to the Calvin cycle, that will get recycled. Okay, so this is CAM pathway. So all the pathways, these are the main points. This much only you need to remember. You can, if you are clear with these steps of this C3 pathway, C4 pathway, and CAM pathway, you can answer every questions from this, because these points are especially from the previous year question papers. So all the confusing points I have explained here. So nothing to worry. It will be easy for you to answer these questions. And next is about photorespiration. Photorespiration or C2 cycle. Okay, see, this is a special type of respiration shown by many green plants. Special type of respiration by green plants. Many of the green plants are showing this type of C2 cycle or the photorespiration when they are exposed to light. When they are exposed to light. And this was discovered by Dicker and Tew. Dicker and Tew. Okay. So first, this is found in tobacco. Dicker and Tew observed this in tobacco, and the site for photorespiration is chloroplast. Site, which site it is taking place? That is chloroplast. Okay, so the site for photorespiration is chloroplast. Peroxisome and mitochondria are also required to complete this process. Here, not only chloroplast, peroxisome and mitochondria are also, these are also contributing to complete this process. So at higher temperature and higher oxygen concentration, higher temperature and higher oxygen concentration, RUBP carboxylase that functions as oxygenase. RUBP carboxylase. RUBP carboxylase works as oxygenase, and instead of fixing carbon dioxide, it is oxidized, or that is oxidizing ribulose 1,5 biphosphate. So here carbon fixation is not taking place. Instead of that, that oxidizes ribulose 1,5 biphosphate. 1,5 biphosphate to produce phosphoglyceric acid. Produce phosphoglyceric acid and phosphoglycolate. Okay, here every reaction you should know what is happening. So this reaction at high temperature and high oxygen concentration, that is the major conditions which are required. So here RUBP carboxylase that oxidizes that, here the ribulose 1,5 biphosphate that is forming phosphoglyceric acid and phosphoglycolate. Okay. And here the photorespiration that involves synthesis of two carbon compounds. Photorespiration that involves synthesis of two carbon compounds. That's why it is called as C2 cycle. Okay, photorespiration is also called as C2 cycle because it is involved in the synthesis of two carbon compounds, glycoxylate and glycerin. These are two carbon compounds, and uh, C2 cycle, this is also called as glycolate metabolism. Okay, C2 cycle also called as glycolate metabolism. This is repeated 3 times in previous year question paper. Okay, 3 times this question is repeated. And here C4 plants, they lack the process of photorespiration. C4 plants, no photorespiration. Or there is no glycolate metabolism in C4 plants. The productivity and the yield in these plants are better than those in the C3 plants. Okay, since C4 plants doesn't have the process of photorespiration, their productivity and yield in C4 plants that is better than those of C3 plants. So C4 plants showing better yield and productivity because that doesn't have the photorespiration. Okay, and next topic or the final topic of this chapter, that is factors which are affecting photosynthesis. There are several internal and external factors which are affecting photosynthesis. Which are the factors affecting photosynthesis? So many factors are there, internal and external factors. External factors are light intensity. Light intensity, we can say initially the rate of photosynthesis increasing with light intensity, but extreme increase in light intensity may have some inhibitory effect on photosynthesis. Okay, when the light intensity increases, photosynthesis increases, but when it is crossing the extreme level, some inhibitory effect also may happen. Then carbon dioxide concentration. That is another external factor which initially carbon dioxide increases the rate of photosynthesis, but later when carbon dioxide concentration is becoming more or it is crossing the limit means that may show inhibitory effect. Then temperature. Temperature after reaching 35 to 40, further increase in the temperature reduces the rate of photosynthesis. 35 to 40, that is a maximum. After this, that will cause inhibitory effect. Above this, then water content. Water content, increase in the water availability will cause steady increase in the rate of photosynthesis. Okay. Then we need to tell about the internal factors. Okay. Chlorophyll content. This chlorophyll content, rate of photosynthesis increases with increase in chlorophyll content. Chlorophyll content increases, photosynthesis also increases. Okay. Then the byproduct accumulation. Byproduct accumulation, after the accumulation or after accumulation, this byproduct will, if over accumulating means what will happen, that will act as inhibitory factor for photosynthesis. That will, if that is increasing, that will work as inhibition or inhibitory factor for photosynthesis. Then the structure of leaf, internal structure, we are saying internal structure of leaf. So that there are multiple factors which affect the rate of photosynthesis, but the extra nature, so the like we can say that exact reason for this is not yet known, but the structure of leaf, that also matters, right? So the rate of photosynthesis, that is also affecting the structure of leaf, and the internal structure of leaf, that also affects the rate of photosynthesis, but the exact, how exactly it is affecting, that is not yet known, but it is also affecting the photosynthetic rate. So here the last topic also we have finished. We have covered almost all the previous year from 10 uh, previous year question papers. From that I have made all the points. So here almost I have covered 10 years question paper to make this point. So it will be very clear for you each and every question from which one. See the site and the pigments involved in the photosynthesis, then the light reaction, chemiosmotic photosynthesis, dark reaction, photorespiration, and factors which are affecting photosynthesis. Factors affecting photosynthesis, that is very simple topic. Okay, but here the light reaction, dark reaction, photorespiration, all these are bit, it will be bit confusing, but now here I have clearly explained everything. Go through that, and in the next class we'll start with a new chapter and all the previous year questions from that chapter. Thank you.